It's always a pleasure to have Linda Griffith visit because she's not only outstanding in what she does as a researcher and as an educator, but she's an alum of Georgia Tech, having graduated with her B.S. degree in chemical engineering, having worked and survived in Ajit Yoganathan's laboratory. Ajit, you must be somewhere here. Okay. But before I say a little more about Linda, it's not often that we have the pleasure of having a speaker's parent in the audience, and so Mr. Griffith, I would like you to stand up so we can recognize you. Okay, so Linda, undergraduate here, Ph.D., University of California, Berkeley, postdoc in some of those lousy Boston labs, but then joined the faculty at MIT, and has had a wonderful career. So she's a great researcher, educator, but also a great friend. Linda, it's all yours.
Thank you, Bob. So 2 introductory things. First of all, about Georgia Tech and its influence on my career. Not only did I have a fantastic experience here as a student that prepared me for a lot of things in life, it's been a huge influence on me all through my career because of what Bob and his colleagues have created here at Georgia Tech. Bob has been an incredible inspiration to all of us in the field. I came up through the ranks as a junior faculty member and had some hard times, and Bob was always there to talk to junior faculty, stimulate the field, pick you up, send you on your way, and I think all of you here are very blessed to be here. It's also been fantastic to see the incredible growth of bioengineering here at Georgia Tech. There was very little research here. I think it was Ajit and one other person really had research labs when I was a student here, and it was in chemical engineering, and there was no bioengineering. So you should feel extremely proud of what you have here now. And I'll also mention I've been very fortunate to have some Georgia Tech students work in my lab at MIT. So most recently, Martha, who's here in the audience, material science student, spent an REU summer, did a fantastic job. Less recently, but very prominently, Manu Platt really came in and shook things up in our mesenchymal stem cell lab, and it's terrific to see him doing so well here. I can't.
He finally got it right.
He's doing great. It's just really great to be back with the. You can't. Oh, the mic is not on. The overflow. How about that?
And for the videotaping, do you want the lights down? Let's see, one way or the other. That's probably better, maybe.
And the second thing is about the talk I'm going to give today and the work, the scope of work that's going to be presented. I'm presenting some things I've never talked about in a seminar. And I, in fact, got data emailed to me last night from one of the postdocs, and with a lot of goings on this week, the site visit some of you know about for the STC. I've been spending a lot of time with Bob Neerum in various locations around the country over the past week. Preparing for a site visit, I had to channel one of my colleagues who couldn't be there. So the talk is not quite as polished as I normally like to have it, but I really wanted to talk about this work because it brings up some issues of signaling and how we control receptors and tissue engineering. And I know there's some people in the audience who are very interested in some of these multivalent effects. So even though it's not completely polished, I felt it would be really fun to talk about this. So please don't hesitate to wave if there's something that's not really clear. And so in my lab we do a lot of basic biomaterial science, protein engineering, as you'll see today, with an eye on some clinical applications. And 3 of them are listed here. Thank you to the folks who came yesterday to talk about the last topic, which is a very new topic for our lab. And the title of my talk is a little esoteric, and another way to think about it is EGFR family gymnastics, in the sense that we want to tame signaling by this incredibly important family of growth factor receptors, the EGFR family, and I'll introduce them to you. Oops, oh gosh, I got to get used to remote here. So there's 4 members of this family. We'll go through their properties a little later. And for tissue engineering purposes, and more recently for us, possibly for cancer therapeutics, we have a tremendous interest in controlling spatial localization of the receptor activation. And so I'll have that as a theme throughout the talk. A primary motivation when we started and this is, I'm going to cover some things that Manu worked on in our lab, is a collaboration with an orthopedic surgeon, George Musler at the Cleveland Clinic. This is one of his patients. The patient had a huge tumor taken out, and typically now there's a replacement process where you put a cadaver bone in it, so you can see that fixed here. Our goals over the past approximately 8 to 10 years of our collaboration have been to come up with alternatives to these kind of cadaver bone grafts by integrating both macroscopic scaffold approaches with microscopic approaches to control the local microenvironment of the cells so that you could combine some source of osteogenic cells with the graft and put it in and have it heal. And the osteogenic cells that are most of interest to George's lab are fresh purified aspirates. So I'm going to give a little background on this just to set the stage of why we got interested in these problems of EGF receptor dimerization. So one approach to healing large wounds, or bone healing wounds, is to take instead, right now, if any of you, has anybody ever had an autograft of bone, like from the hip? So now they typically will take autograft of bone for the hip, which has, harvesting that bone actually has its own huge morbidity issues. So there's a cadre of, small cadre for now of physicians who are trying to use marrow that can be aspirated intraoperatively. So instead of actually taking the bone out, they just harvest marrow. And George has optimized some methods to do this intraoperatively. So the gist of it is that when you harvest marrow, about 1 in 30,000 of the nucleated cells have properties of very early osteoprogenitors and maybe true stem cells. So there's a mix of cells in these marrow aspirates that can go on to form fat, cartilage, and bone. And for today, we're interested in this. Okay, and I'm being very careful to call these progenitor cells and not strictly stem cells, because you always get a mix, and the assays we do not guarantee stemness, despite the popular use in the literature of mesenchymal stem cell as a term. It's been shown that if you take that marrow and pass it over a graft, demineralized bone, etc., as is shown very crudely here, there's actually a machine now that does this. So you pass it over the graft, the stem cells can be selected, and this can be enhanced bone wound healing. And so George has been very interested in this idea that passing the marrow over a scaffold, and a couple of them he's used are shown here, by mechanisms not well understood, you can enhance the progenitor cell concentration and prevalence in these grafts. Okay, and this is work that's been published, and so we've been interested in figuring out how to do this and how to actually use it in the clinic. You don't always have to understand everything before it's useful in the clinic, right? And it's iterative. Okay, and so together we've worked on building scaffolds that can entrap these cells, working together with Theryx, a company that has commercialized this 3D printing process to make scaffolds. So we designed scaffolds that would, we hope, trap cells, and in fact showed that the scaffolds that we designed will trap these connective tissue progenitors as well as demineralized bone matrix. And as we think about translating these things into the clinic, though, there's an issue of this microenvironment. If we're going into a really large defect where there aren't that many stem cells, we may need every stem cell to do a huge job. So the problem we face is that different patients have different prevalence of stem cells. So these are the same exact number of nucleated cells from 5 different patients that are plated in what's called a colony assay. So you plate the marrow, 500,000 nucleated cells. The cells that attach and form colonies are ones that are believed to be the progenitor. So you can see the incredible diversity in prevalence. So this patient has very few, and this patient has a gazillion of them. Okay, and again, you can see the heterogeneity in colonies. And another area of interest in the lab, not one I'll talk about today, is building better colony assays. Okay, so the challenge is now these cells are going to go into this really hostile microenvironment, right? There's all kind of, it's hypoxic, it's filled with cytokines, and some of my slide is cut off, but we'll live with that. It's filled with cytokines. If you have an excess of stem cells, like if you're doing a transplant in a mouse, you may put in 100 times more cells than you need. So you'll see, bone grows, okay, even though maybe a lot of the cells are killed. But in a human defect that's large, you may only have close to the number you actually minimally need. So we have to worry about whether some of these cells are going to be killed. So we started thinking about ways that we could influence different steps in this process. And in particular, if we look at a stem cell, a progenitor going to a bone-forming cell, there's a lot known about different factors that act at different stages of development, okay? And in particular, bone morphogenetic proteins are widely used now in the clinic to facilitate bone healing in spinal fusion, in open tibial fracture repair, and a few other applications. Parathyroid hormones used also to stimulate bone for osteoporosis. But these really drive differentiation of committed progenitors, and they don't necessarily cause proliferation of those progenitors. And so if we drive, our hypothesis is that maybe approaches in these large defects have not been successful because we drive premature differentiation. What if we go further back and try to drive proliferation of this compartment and yet also influence downstream processes? And so there's a number of factors known to act here. One of them, interestingly, is epidermal growth factor. Its signaling by EGF receptor is involved in many stages of bone development and homeostasis and healing. So as boring as many people think EGF receptor is, first growth factor discovered, there we have it as an interesting molecule. So our question is, can we stimulate the survival and growth of these cells that we transplant using EGF? It's attractive, it's been around a long time and we know a lot about it, but it's never really been developed into a successful clinical product. And part of that, as we started thinking about this many years ago, started thinking, why didn't Johnson & Johnson or anybody actually ever have a clinical product? And I'll come to that in a minute. It turns out there is actually a commercial product. If you inject soluble EGF into sheep, it circulates with a half-life of a couple of days, and the sheep, when the hair grows out, it's very thin, and you can actually pull the wool off the sheep, as you see here. And in Australia, kids on the farm, kids don't like staying on the farm. Sheep shearing is very skilled, so having this is actually a now commercially viable operation in Australia. There's also, if you go on eBay, Google EGF, you can find these facial anti-aging products. They cost like $200 for a little vial you can barely see, and it says FDA doesn't prove that these work, but I'm 85 and I use these products, so you can decide for yourself. Okay, so our hypothesis, though, we went back and we looked at, so let's look at physiology of EGF receptor. There are a lot of ligands for this particular receptor. It's, for those of you not familiar with it, it's got an extracellular domain that binds one of several ligands, a transmembrane domain, and an intracellular domain that transmits signals. And we'll come into this part more later. And so it activates, it's a receptor tyrosine kinase. It initiates several cascades of signaling through its tyrosine kinase domain. So we asked the question, most of the ligands actually are somehow restrained in the extracellular matrix and may actually signal in a more tethered fashion than in a soluble fashion. So we started thinking about this concept of delivering EGF from the matrix, and this brought in some design issues because the EGF receptor has to dimerize in order to signal. And there is some controversy still remaining, but the evidence is overwhelmingly in favor of the receptor kinase domains needing to come together to initiate phosphorylation cascades. So there's been crystal structures published of the extracellular domain in complex with the ligand. This happens to be TGF-alpha. And so one could imagine tethering this molecule via its N-terminus, there's only one primary amine in murine EGF, to a substrate. And so we initially decided to do this using polyethylene oxide, very simple approach, and space these so that we could get clustering and dimerization of the receptor. So in work that actually Darryl Irvin contributed to during his thesis, we designed a polymer system that would let us present little clusters of EGF receptor on the surface or other ligands, shown schematically here, hydrophobic backbone, polyethylene oxide side chains. When you cast these on a surface, they make these semi-interpenetrating discs, so you can make little clusters of ligands by mixing modified polymer with unmodified polymer and coupling here. So you can get a structure like that. And so then if we want to examine how this affects MSCs, we could speculate that it will initiate signaling cascades because EGF binds at the surface, dimerization occurs, and then typically the receptor and ligand are endocytosed and trafficked through intracellular compartments, but signaling is initiated at the surface of the cell. So you have EGF receptor dimerized here, initiating several different signaling cascades, PI3 kinase, ERK, and PLC gamma, which involved in motility. So one question we speculated is maybe tethered EGF would actually act better than soluble in protecting cells against pro-death signals. So tethered EGF would stimulate the receptor, protect against pro-death signals, because these signals tend to be preferentially on the membrane. And so in work that Vivian Fan did in her thesis, we looked at whether tethered EGF had any effect on protecting cells in the kind of pro-death environment they would experience. So these are primary human mesenchymal stem cells. We get our cells from Darwin Prokoff's facility. He's now in Texas. These are very low passage 3 or passage 4 cells, but they're called mesenchymal stem cell or merostromal cells, we'll call them. So no treatment. If we add Fas ligand, we kill the cells, but if the cells are cultured in the presence of tethered EGF, in fact, they look protected. And in quantifying this, if we look at the percent of cells that are dead, this is the control, so very little cell death, okay? Add this Fas ligand, pro-inflammatory or pro-death cytokine, and you see a lot of cells die. Soluble EGF actually potentiates this, and this we think may be due to interruption of autocrine loops. We're investigating that now, but tethered EGF protects against that cell death. So that is interesting because it's an important way we might influence the survival of these cells. And so we went on and asked, well, does it enhance colony formation? And here, this is again from Vivian's work, just showing plating cells early on, soluble EGF compared to tethered. Tethered actually enhances adhesion, probably through a localized actin polymerization going through NWASP cofilin pathway, for those of you who love the signaling stuff. But you can see dramatically it enhances, and it's actually dependent on activation of the ERK signaling cascade we showed. So then Nick Marcantonio, in his thesis work, investigated whether primary cells colony formation from marrow plated would be enhanced by this molecule. And in fact, what we found, looking at different adhesion substrates, an environment where there's only adsorbed serum proteins, one where we used a synthetic RGD that has the synergy site from fibronectin, or fibronectin itself, so those are our 3 adhesion backgrounds. Now we add tethered EGF, and in each case we see colony formation is enhanced. So with tethered EGF, with tethered EGF, with tethered EGF on fibronectin, interestingly, soluble EGF inhibited compared to the control for fibronectin. So there are effects on these cells, and another student in the lab, Lou Alvarez, who just defended his thesis, went on to say, let's see if we can develop ways to put these on to scaffolds that are clinically used. So the company that's commercialized three-dimensional printing, Therix, has a line of products based on tricalcium phosphate, and this is just showing pictures of it. So they do very well in the clinic, and this is one substrate we envision using either alone or in combination with a polymer. So Lou, working with some undergraduates in the lab, developed an approach to tether EGF to this substrate, and I'll talk more about the protein engineering later in the talk, but the conceptual part here, we used phage display to identify a peptide sequence that binds with very high affinity to tricalcium phosphate, and this is just some data showing that there's a specific binding, and then EGF is here, and you can just view this for now as a spacer. Okay, so he's got this, he puts the scaffold in a solution of this molecule, it absorbs on the surface, and now we've got tethered EGF, okay? So if there's no binding domain, you don't get binding. So here, there's specific binding. Here, there's none. So it's pretty tightly immobilized on the surface. And this is just showing the construct. You do phage display. We got a consensus sequence shown here using several different kinds of substrates for screening. And this is a manuscript that's in preparation from Lou Alvarez's thesis. Okay, so now we take scaffolds treated with tethered EGF and look at the effects of this tethered EGF on primary mesenchymal stem cell proliferation. And so what we see is that, and this is just looking at a number of cells, what we see is by day 7, this is in serum-containing media. So you've got all kind of growth factors there. And even in serum-containing medium, you can see there's a tremendous increase in cell number at this early time point, and even at 14 days, there's statistically significant increase. And also in serum-free medium, we can see that these enhance survival of the cells. And Manu has a couple of papers that came out that also investigate further signaling properties of tethered EGF in our original format. And I'm sure he's talked about those here. But a basic question comes up. Putting EGF on the surface in this way, we wouldn't expect it necessarily to be close enough to dimerize very efficiently. So there's a stochastic presentation on or near stochastic. So we can ask the question, is this really acting through an EGF receptor? We can inhibit the EGF receptor kinase and abrogate the effects I just showed you. So we're convinced it's acting through EGF receptor, but is it acting through homodimerization is really the question. Because we would predict that there wouldn't really be that much completely correctly positioned to homodimerize. So now we're getting into this question, why would we get this signaling? So let's go back and look more carefully at the EGF receptor family. I have a couple of slides in sequence to take you through because it's a little bit complex, and I took a picture out of the literature and let me take you through it. So there are 4 family members. This slide is showing ligands for the family members. It's showing the receptors and the various kinds of dimerization they can ensue, downstream signaling pathways, and transcriptional control, okay, that influence these things. So it's a comprehensive, one thing that's a comprehensive picture. Okay, so what you see here, if we blow it up, is that EGF receptor can heterodimerize. It can homodimerize, as shown here, but it can also heterodimerize. Notably, yeah.
I think your mic went out. Let me just
Oh, okay. Okay, this was the one that Bob's going to use to shock me and keep me awake. It's been trying the whole time here, and I haven't been jumping around.
So obviously it wasn't working.
Yeah, obviously not. Wait, no, you can't move this one. Take the bad one. Okay. Part of why we focus on this is I have this whole other story in liver cells where they only have EGF receptors. But mesenchymal stem cells actually have HER2. HER2 is the famous. Are we done?
No, we're not done.
HER2, can I talk to them? HER2 is the one that's not important. So anybody who's heard of Herceptin, the anti-breast cancer drug, it targets HER2. HER2 is overexpressed in mammary tumor cells, and so it's a very potent signaling. And HER2 does not have a ligand, so HER2 has to partner with someone. HER2 has to join up with EGF receptor or one of the other ones, and it loves to join up with EGF receptor, can also join this guy. So it has to partner with someone to signal. And EGF receptor can heterodimerize. So mesenchymal stem cells have HER2 and they have HER3. Okay, they don't have, there's another guy, HER4. They don't have HER4. Okay, so they have all of these. So these are potential partners that you could imagine are present in MSCs. So it's entirely conceivable that our MSCs, when they bind monomeric EGF, monomeric EGF, they're making these kind of heterodimers. Okay, and these are very potent in signaling for survival. So that could be what's happening on those scaffolds I just showed you. Okay, so we could then say that monomeric EGF is acting through these heterodimers, and it raises a question, what if we don't want those heterodimers to form? Actually, what if we would imagine in cancer cells keeping HER2 from joining up with HER1? What if we could pull HER2 out of the party by forcing these two receptors to homodimerize? Okay, so that's one interesting thing. So in other words, here again, just showing it a little cleaner, we've got EGF receptor HER2 heterodimer, EGF receptor HER3 heterodimer, and showing HER3. So this is MSCs, and then they crosstalk with integrins. We bind EGF. EGF has very high affinity for EGF receptor, not so much for HER3. And if we want to completely manipulate this entire family, we can look at ligands for HER3. Okay, HER3 can also heterodimerize with HER2. Okay, HER2 doesn't have a ligand. That's supposed to be a blank. So neuregulin is a molecule that binds here. Okay, so if we think about manipulating this system, we might also be able to keep HER3, HER2 heterodimers from forming. Basically, the name of this game is exclude HER2, keep HER2 from coming to the party. Okay, so this is a schematic. So if we come back now, we can say we want to control this dimerization, and how could we do that? So the idea is, could we take a cell that expresses multiple receptors? If we give it monomeric, we have no control over the dimerization partners. But now let's make a bivalent ligand. Let's make a ligand that you tether the two together, and now can we bias dimerization so that we only get a particular receptor pair? Okay, now you can imagine this may not work because you may get these to act just like a monovalent ligand, right? One might bind and then the other thing is free. There's a lot of things that can happen, so we have to test it. So we've made a family of these dimers, and I'll show you how in a moment and show you some data. So here's the receptors. We have EGF and neuregulin are our main two players, and then we can make various combinations. Ligand dimers, EGF, neuregulin, neuregulin, and theoretically at least drive different kinds of dimerization, hopefully to the exclusion of the heterodimers we don't care to form. Okay, in order to design these, there was a fair amount of protein engineering that Lou Alvarez did, and this was in collaboration with Rich Lee, who's at Brigham and Women's. And again, starting with the crystal structure, thinking about these many design issues, going in and looking at the structure of the molecule. So this is EGF, showing the 3 disulfide bonds. There's a lot of information in the literature about EGF receptor interacting with its ligands because it is such an important system. And so we know what will likely happen if we manipulate the various ends. They're not really involved much in binding. And we also know a lot about the affinities. So we could go in and say it's probably okay to manipulate either the C or N terminus. And in fact, we went in and showed it doesn't really matter. You can manipulate these. So we designed a facile system to let us look at combinations. This wouldn't necessarily be what we'd use in the clinic, but it made it very easy to do experiments. There's so this is the essential design shown here. A coil-coil domain is used to link 2 different ligands together. This coil-coil domain was published. It's got a very low picomolar binding affinity, and we verified that there's a very high affinity when we make these ligands. So now we have this coil-coil domain, and here we can actually link something to it that would let us stick this on the surface. And actually we could do a biotin, we could do the TCP binding protein, etc. So this is a place that we can stick it to the surface, or we can use it in soluble form. So this is simply the design of the construct. So the coil-coil are held together. This is a spacer region designed by Bob Sauer's lab that is very resistant to protease degradation so that we can keep that intact. Okay, so we go in and we characterize. The first thing we do, we make these ligands and ask, are the ligands in their monovalent form similar to those that are the native, the thing that you would buy from Preprotech, or collaborative research. So this is EGF that you buy. This is our ligand, and we do the same thing with neuregulin. And go in, and in every case we can activate. These are HeLa cells, which have approximately the same number of EGF receptor and HER2, about 50,000. MCF7s have HER3 and HER4, predominantly with some HER2. So we can activate with all of the individual components and inhibit. This is with an inhibitor of the EGF receptor kinase, or specific kinase. Okay, so these just prove to us that these are active. Then we can go in and characterize this binding. We did a whole lot of different kinds of assays. This is simply showing one, which is a binding to a surface, and we get about an 8 picomolar affinity. Isothermal titration calorimetry experiments verified this in a number of different Biacore measurements, very high affinity binding. So at the concentrations we would use them, they're essentially all dimerized. There's very, very little free ligand. What I want to finish up the talk with is 2 things: some phenotypic data that has gotten us excited about this, as well as some signaling data that are starting to explain the phenotypic data and supporting our idea that we are forcing dimerization of these receptors with these ligands. Okay, and we predominantly have used actually 3 cell types. One's not shown here. these mesenchymal stem cells. What's notable about them, they have very few number of receptors. So about 10,000 EGF receptors, a few HER2, and this is work Manu characterized during his postdoc. They do have HER3, but no HER4. MCF7 cells are a very widely used breast cancer cell line, and they express a lot of HER3 and HER4 and very little EGF receptor or HER2. And HER3 and HER4 are harder to characterize numerically just because of reagents, but this is what they do. We also not shown, as you saw a moment ago, use HeLa cells. HeLa cells are, and also primary cells. So HeLa cells are a cervical, they were actually the very first widely used cell line, and they have EGF receptor and HER2. Okay, so let's just look at some phenotypic data, and this is serum-free culture of a mesenchymal stem cell line. It's immortalized with hTERT, human telomerase, and this is culture over 23 days. And so these are hTERT immortalized. hTERT is pro-survival and inhibits some of the apoptotic pathways. So even when you don't add serum, these are plated in serum, allowed to establish for 1 day in serum, then we take the serum out. So there are cells that survive, even though, they're plated at high density, they'll survive. These are really, pretty tough cells. Okay, if we add EGF, there's actually the quantitative data show that there's not quite as good survival of these cells. It's kind of a little bit hard to see here, but there's a little bit worse actually than here. And that could be because EGF may be inhibiting autocrine loops that are pro-survival. We have talked about this with Manu. Interestingly, the EGF receptor homodimer, you see really terrible, so in fact very few cells survive. So there's some huge difference between these guys. Okay? Neuregulin is, manna from heaven for these guys. You could see they're incredibly able to survive in the presence of neuregulin. And HER3, interestingly, HER3 doesn't have a kinase, so it can't act by itself. But when it heterodimerizes with HER2, it sends a very, very strong signal through the pro-survival AKT pathway. So this is consistent with what we would expect from a pro-survival signaling. This homodimer, or heterodimer, EGF and neuregulin, also pro-survival, but interestingly, so this is native neuregulin, this is the dimer, very poor survival. Okay? These results are actually what we would predict based on, if we were forcing EGF homodimers, we'd get worse survival because we've taken HER2 out of the party, and HER2 is a more potent pro-survival signaling. Here, if we've taken HER3 out of the party, HER3, we're shutting it off, again, we get poor survival. So it actually is consistent with what we expected. Another very important phenotype, both for tissue regeneration and cancer, is migration. And we follow this a number of ways. One is just simple 2D migration experiments, random cell motility, which you can follow with time-lapse video microscopy. This is an image that shows, taken by Kevin Korn, a summer REU student from UT Austin in collaboration with Edgar Sanchez, who's currently writing his thesis proposal in my group. And so what we have here is you can see these cells, so this is a single field, and this shows the trace of where the cell wanders during the 12 hours we observe it. So the cell starts here and wanders here, the cell starts here, wanders over here, and then there's various things that can happen. The cell can divide, it could die, it could wander off the screen. A few % of cells have these kind of weird events, and we take them out. They don't statistically change the migration analysis that we do. Okay, so we can do these kind of experiments on a lot of cells, and this is a little bit faint, but the tracks didn't show up so well. EGF has pretty much the behavior that was shown here on, the tracks are not showing up so well. So you can see these guys are very, very active. But when we have the neuregulin, the cells are just spinning around in their little things. For some reason on this screen, we're not really seeing it. If we summarize, there are different migration behaviors induced by the ligands. So essentially, some cells hardly go anywhere. They spin in circles, or they just are not motile. And this is observed particularly with these ligands here. Then phenotypes where the cells move really fast and really persistent are observed in other conditions. Okay, and I'm going to try to show you some data, not a very pretty slide, but one way, there's a lot of different ways to interpret migration data, and we're sorting through how to compare different modes. But one way is just to say what fraction of your initial population moved total more than 50 microns from where it started. And so if we look there, and this is the concentration of the various ligands, if there is nothing there, just by happenstance, about half the cells move 50 microns. That's just by happenstance, it happens to be 50%. If we have EGF, there's an interesting kind of slightly biphasic behavior. It's similar, to none, but then we start to inhibit migration here. The homodimer, tremendous, first it inhibits at low concentrations. There's a tremendous biphasic effect where it's very stimulatory here and then comes back down. Neuregulin is inhibitory at low concentrations, starts to stimulate as we go higher and higher. And the homodimer, though, completely opposite behavior. So this is neuregulin, this is the homodimer. It's essentially shutting off signaling. And so the point of this slide is to show you that quantitative effects are very different when we have a monomer ligand versus a dimer ligand. So again, monomer here, dimer here, really opposite effects. Okay, what might be going on? Okay, and then another kind of assay is chemotaxis in a transwell assay. So this is a data not using mesenchymal stem cells. This is what has traditionally been known as a breast cancer line. It's actually a melanoma line. And this was from Jeff Siegel's lab. He has some HER2 overexpressing cells. So the HER2, HER3 heterodimers are very potent for invasion. So they're very metastatic cancer cells in vivo models. And now if we take these cells and set them up in a chemotaxis experiment where they're undergoing chemotaxis to neuregulin, which is something they normally love to do. They love to migrate to neuregulin, and we add a constant concentration of our dimer to both sides of the assay. We can see that the neuregulin, so there should be, the dimer concentration when there's nothing, you get migrate. These are actually, dimer concentration here is, let's see, so when there's nothing, you get really great migration, but the neuregulin, these are with no chemotactic agent added. So these are sort of our controls. The E got cut off here, so this, or the neuregulin got cut off. So this is with no neuregulin present, so this is our control. There's no chemotaxis essentially. If we add now the neuregulin, we get chemotaxis without ligand, so this is chemotaxis without ligand. The neuregulin homodimer completely shuts that off. So it's consistent with our hypothesis. Now, why might that be happening? In fact, if we're now going to start looking at signaling, for example, activation of ERK as measured by phospho-ERK, we can see neuregulin activates ERK, okay, in a dose-dependent manner. Bivalent neuregulin, very modest activation, and as you go to higher concentrations, it shuts it off. So this is completely consistent with the view that we're forming these homodimers of HER3. HER3 doesn't have a kinase, so the homodimers just sit there. They can't do anything, and so nothing happens. Okay, that actually could be very good for cancer cells. Okay, so let's think about an experiment in these MCF7 cells, which actually have all of the family members. Notably, they have HER3 and HER4. So we're going to have to do a kind of tricky experiment with this to observe it. Again, our HER3 homodimer shouldn't signal, but neuregulin binds to both HER3 and HER4. So in this experiment, we're going to do some controls where we block the HER4 axis by using just an antibody that binds to HER4. Okay, so we're going to if we add, these don't signal, this would signal, okay? This would signal, but this wouldn't signal. Now, HER4 dimers can form, so we can block that if we add anti-HER4. So if we add anti-HER4, we knock this out, and this is silent anyway, so we should be able to block. Okay, so we do the experiment. Here, there's nothing added here. We add an anti-neuregulin antibody to block HER4. We put in our ligand, okay? So then we put in our bivalent neuregulin, and then we stimulate with neuregulin, okay? So if everything's hunky-dory, the neuregulin will cause the cells to signal, right? Okay, but if the bivalent is inhibiting, we'll see a dose-dependent inhibition, okay? So in fact, again, this is just showing a native neuregulin. You stimulate ERK, it's highly stimulated. Again, they have both HER3 and HER4, so this is completely expected. Okay, now if we do this experiment, okay, we stimulate with, we add either plus or minus the antibody. We then put our bivalent ligand in, and then we stimulate with neuregulin. And this is a dose response. So at very low doses, neuregulin is able to stimulate these cells. Okay, so neuregulin's doing great. And now, as we continue to add this, higher and higher concentrations of bivalent ligand, we shut off, again, pERK signaling. So this is if we inhibit HER4, if we don't inhibit HER4, and this is kind of expected you would release it. So it looks like in these cells, we are in fact shutting off this axis of signaling, and in fact, it causes cells to undergo apoptosis. When we do an apoptosis assay, percent just dead cells, when we go in and again look at a phenotype. So this is actually pretty exciting because it offers an opportunity, a different way. I mean, who would think to shut off the signaling by this terrible, in a lot of cancers you've got this HER3, HER2 heterodimerization that's causing a lot of problems. Antibody therapies are very difficult because antibodies are big and they actually have a hard time getting in tumors. What we're finding with this, these are very small molecules. So there's potentially a lot of therapeutic potential. Who would think? Give a ligand for what you're trying to shut off to shut it off. So my lab isn't a cancer lab. We're working with some other labs now to pursue this. Okay, just to finish up real quick on the MSCs, the signaling is sort of the same story. I focus there on HER3, HER1, HER2 heterodimers. Again, we can control the signaling or drive this, the signaling data in terms of if we have bivalent EGF, we can get dose response, a relative increase relative to monovalent and EGF receptor phosphorylation, but we see a decrease in HER2. So the signaling data shows a shift into EGF receptor that we'd expect. Okay, so what I tried to do today is present you a motivation for why we went down this path of this protein engineering, looking at receptor dynamics, some initial data that this could be a very interesting way to manipulate this receptor family, and I'll close with one of Yossi Yarden's really nice photos of the complexity of this pathway all the way from production of the ligands by shedding, proteolytic shedding from the cell surface, binding, dimerization. Receptors can even be cleaved, influencing all these pathways. What I hope we've done is not only develop things that may be useful in the clinic, translational therapeutics, but also as tools to help us sort out what some of these signaling pathways are in the EGF receptor system. I have tried to mention everyone involved with the work. Rich Lee was Lou's thesis co-advisor. It was mainly the work of him. Seymour, who was from EPFL, Jeff Hubble and Melody Swartz, sent him over to do his master's. And Linda Stockdale is our technician who makes a lot of the polymers. Edgar Sanchez did the video tracking, and Edgar's on there twice. I guess he did a lot of work.
He must have done a lot of work.
Yeah, he did do a lot of work, actually, for his undergraduate.
Has he got 2 degrees?
Well, we have 2 tracks. Yeah, maybe we can give him one in both tracks. Okay, so wanted to share with you just some thoughts about this family, and be happy to answer any questions.
Okay, it's open for questions, comments.
What? I'm trying.
Linda, I enjoyed your talk very much. With this dimeric ligands that you can have the N or the E, so very interesting system. Have you tried a simple experiment in which you IP for the ligand and show that you can pull down both receptors?
We're doing that right now. Part of, as you know, different research projects take on a life of their own. The mesenchymal stem cells have so few receptors that the initial attempt to do that was not very successful. So it meant bringing up some of these cancer cell lines where there's a higher receptor expression level. So we're in the process of doing that, and particularly because they have a biotin linkage site, so you can put that on. So Robin Prince, who unfortunately her name's not here, she did the chemotaxis experiment. She's just doing a short-term postdoc to sort out those issues. So we're in the process of doing that. Those experiments can, they can have a lot of artifacts, so it's tricky to get them to work right.
Your ligand is so clean that you should be able to do it.
We should be, and particularly because we have that biotinylation site. So Robin's in, and that's actually one of her projects right now is to actually get that. Another thing we're doing, and as part of the STC, is we're labeling both of them with chromophores that are non-fluorescent and become fluorescent when they bind, and this is Barbara Imperiali, so that we could actually measure the binding by increase in fluorescence upon binding of the ligand, and so we just got a postdoc to start on that. These methods to prove the physical association are really tricky to get to work, as you No.
Yeah.
Yeah, they're really tricky to get to work.
Okay. Questions?
And I'm happy to stay and talk informally to anybody afterward. Yeah.
Thanks so much, Linda. Have you looked at all about changing the spacer length to see what that might have on the transduction that occurs?
No, we haven't done that. We based our spacer length off some work that we had done with the comb system and what we thought was effective. But that's on the docket to do. In fact, the new postdoc that we have with Barbara's lab, she's got a method. What we really want to do is not have them joined by the coil, but actually have them as one protein. If you express just one protein, though, you get a giant mess because the each has 3 disulfide bonds, and you get a wad of crud. So what you can do, though, is you can make each one separately and ligate them. And this is just not stuff I do in my lab, but Barbara Imperiali's lab does. And so she's going to, well, neither of us are going to do any of this. It's Andrew Kruger who's going to do it all. Andrew is going to do exactly that, make a series of systematically varying spacings, ligate them so we have a single molecule proteolytically resistant spacer that we could do exactly what you said, because those would be very interesting. I tend to be, there's a fine line between doing a lot of biophysics and focusing on the phenotype. So, but this is biophysics. It's not that hard to do, and that we should do. So we will. Great question.
Other questions?
We haven't done experiments like that. I think partly again, tend not to think about activate because they have to be activating antibodies. We use the MAB225 very frequently to inhibit binding of ligand to EGF receptor. In fact, have some data showing that we can inhibit the effects we see with the dimer if we use a MAB225. But I guess we could, you could do the same thing with a anti-HER3, but we haven't done that. I'm not sure what it would say exactly. To think about that one. Yeah.
No.
Yes, very nice talk. So in the first few slides, you showed an experiment where you had tethered EGF, so you had a lot of EGF on the surface.
Yep.
That seemed to be promoting cell survival. Then at the end of the talk, we had the dimer that seemed to be killing the cell.
That was in solution, and if I wasn't, none of the final stuff, none of the dimer work was tethered. And
well, why did you see a difference between cell survival, pro-survival on a surface versus pro-death in solution?
Okay, so there's a lot of potential things that could be happening. So first of all, in solution, solution things can be internalized. What we haven't done yet, or done a very good job of with data that I would show here, is look at receptor trafficking. So if you give a soluble, you can downregulate the receptor because it's internalized and degraded. If you give tethered, you inhibit that. It turns out you don't completely shut it off because all of this takes place against a background of autocrine ligands. So the big frontier now is to understand how autocrine ligand secretion, and this is, Manu started down this path, it's a very sticky wicket to characterize all these things, but the cells are making at least TGF-alpha, definitely amphoregulin, so they're making autocrine ligands. So now, some of the EGF receptor ligands, if they signal from the surface primarily and you retain receptor expression, they can be pro-survival. But some ligands cause receptor downregulation. So if we give soluble EGF, the hypothesis we have is that we're downregulating the receptor so that the cells now can't even respond to their own pro-survival ligands. So if they're making amphoregulin tends to be pro-survival, probably because it inhibits internalization, or it's way more slowly internalized. So these are issues that really EGF receptor family aficionados are probably interested in. But the big picture is it probably changes the dynamics of availability of receptor at the surface and thereby allows signaling. So the tethered, you're keeping it at the surface. You're also HER2, HER1 heterodimers traffic way more slowly than HER1. So you're keeping it at the surface is one explanation. You keep receptor at the surface, and you have.
It's always on.
So it's hard to say it's always on because you do have phosphatase. There's a lot of ways you can desensitize it, and we have tools now. The point I want to make in the last slide, there's a lot of complexity in this system. We're building tools that will let us dissect out some of these effects.
Okay, any other questions? If not, I think we should give Linda another hand.
different